Good morning, everyone. I'm Tabea Eimer, a PhD student in the Health Psychology Group and Lab for Gut-Brain Axis Studies at KU Leuven. And today I'll be presenting a pilot study that we conducted on cross-organ comparison of interoception. Lucky for me, that was already introduced this morning, so that makes it a bit easier for me. So what we see in the literature that interoception is often assumed to be this general ability that people have, but it's not really been assessed if that's actually the case. There are some studies that have ran correlations using the tasks that are out there to assess interoceptive accuracy. Results seem to be mixed. It looks like most of those find no correlation, but the issue we see with those studies is that the tasks that they use to compare interoception across domains are not really comparable in itself because they differ in response requirements or intensity levels or stimulation characteristics. So we wanted to look at, is interoception a domain-specific or general ability using tasks that are as comparable as possible across organ domains. And we compared interoception across the cardiac, respiratory, and gastrointestinal domain. The idea here is that we have in each of those domains the possibility to present different levels of stimulation. So in the cardiac domain, we asked participants to cycle on a cyclogometer, and we can manipulate the resistance, so the difficulty of how participants cycle. And then in the respiratory domain, we used inspiratory resistive loads. For those of you who don't know, these loads can make it more challenging to breathe in. And again, we have different kinds of loads that we can present, so different stimulation levels. And then in the gastrointestinal domain, we used an esophageal balloon catheter, which we insert through the nose into the esophagus, and it has a balloon at the end that we can inflate to different distensions. Now, this was within design, of course. All participants went through all 3 organ domain assessments, one after another, not at the same time, to be clear about it. But we have similar tasks that we use in each of those domains, and I'll be presenting those at the same time. There were more tasks, but I will only be presenting those 2. So in the beginning, we wanted to first check, because perception differs so much across participants, we want to find an individual maximum that participants would have. So we started with a very low stimulation in each of those domains, so low level of cycling, low resistance, low balloon inflation, and low inspiratory resistive load. We went up level by level until they indicated that their maximum was reached. And in this part, one stimulation would be 2 minutes of cycling, and then 1 breath, 1 inspiration, and 1 inflation, 1 balloon inflation. And then we went on to the next task. Of that maximum, we calculated certain percentages, 10%, 20%, 30% of the respective maximum from that participant. And we went up to 80%. So we had 8 different stimulation levels. And here, 1 stimulation would always be 1 minute of cycling, 1 breath, and 1 balloon inflation. And we stimulated and asked participants after each stimulation for an intensity rating of that respective stimulation. Maybe also to mention here, in the respiratory domain, we did flow targeting to sort of control a bit for how participants were inspiring and that they wouldn't change it respective to the load that they were receiving. Now, the sample was healthy young adults with 33 participants, and the mean age was just below 20. What we see is that, so the way we quantified interoceptive accuracy is to look at the change of intensity rating over change of objective stimulation. And we calculated basically a regression for each participant at each organ domain, and we took the slope parameter as an index for interoceptive accuracy. So each participant has this regression slope for each organ domain, and then we correlated those. What we can see here a bit is that in the respiratory domain, the range of rating seems to be a bit higher. That's one of the reasons we did the pilot study, was to really check if the tasks are as comparable as possible. Ideally, we want the range to be sort of more similar, and that's one of those adaptations we'll be making for the next study. So now this is all of the means in one plot. The correlations of those duos show no significant results, which does point into the direction of interoception. Does not appear to be a general ability. Certain limitations we still have, looking at how comparable is this assessment across domains, is that we have a difference in invasiveness. You can imagine it's a lot easier to just cycle on an oculomotor in the lab as opposed to having a catheter inserted. But then also the level of feedback through the stimulation. When participants cycle on the oculomotor, it's more this indirect increase of the heartbeat through that, but just by cycling, you already notice if it's easier or more difficult to cycle. So you have more feedback as opposed to the catheter in your esophagus. You either feel the stimulation in your esophagus or you don't. I mean, there's no external feedback you have from that. So those are the limitations that we have, but on other things, we can still make some adaptations for the next study, which is going to be to investigate the same in a bigger sample. So we will be doing the same tasks, but also to add EEG. Specifically, we want to look at evoked potentials in this case and assess interoception across domains, looking at EEG as well. And with that, I want to thank the entire project team. It's many people involved, especially I want to shoot out a thanks to my supervisor, my main supervisor, Andreas von Leupold, and all of you for listening. Thank you.
Such a cool study. Thanks, Tabea. Any questions? Marcia.
Thank you, really great talk. Did you look at, or do you plan to look at how much all this rhythm lock into each other at different loads, and how much this locking can predict whether you may perceive it better or not, each of this rhythm? Or in other words, I have in mind that when we are a little bit sleepy, for example, respiratory and cardiac rhythm tend to be locked. You have 3 heartbeats per respiration cycle, while if you act or you are engaged into some action, this locking tends to be more variable. And therefore, might be the case that when you ask somebody to perceive your heartbeat or your respiration, if they're locked, you can actually take advantage of more than one rhythm, where if they're not, then you're a little less skilled. Yeah. And I guess that across these different loads, also you may look at this different locking and see whether this can help you to see whether the interoceptive accuracy change.
Mm-hmm. That's a really good input, actually. I think for now that wasn't the idea to look into, but I think certainly since in the next study we'll have a much bigger sample, more power to look at things, and the EEG signal, of course, that's certainly something we could look into. Yeah.
I would also have a question. So did I get it right that your correlation in the end, so you measure whether there was some link between interoceptive domains, was dependent on the slope that you find in the intensity ratings?
Yeah.
Okay, so one other explanation could be that you assume a linear increase, a linear relationship between intensity and so the rating and the actual intensity. That could be the case for one domain, but not the other. So not necessarily the interoception would be different, but just the relationship between stimulation and evaluation, right?
Yeah.
So is there any way to account for that just in the way that you set up the experiment and not make let's say an overly conservative inference on what people are able to perceive?
Yeah, that's actually a really good point. Especially in the esophageal domain, we struggle a bit because the lower inflations, the lower percentage, let's say, don't necessarily inflate the balloon. You can imagine if you blow up a normal balloon, you have this certain threshold that you need to overcome, and that's what we see in the ratings as well. In the beginning, ratings are lower, and then it goes up. I think looking at the smaller sample size now, we might also not have enough power to do more sophisticated analyses, but that's certainly something to look at later as well. For respiratory and cardiac, it was more linear, which is what we like to see.
Also, random intercept might help, right? To just balance the...
Exactly. Something, again, we may have not have the power in the sample size to look at, but yeah.
Could be enough. Thanks, Tabea, again.
Thank you.